Good morning. So thank you for coming to the last morning of our conference. This is a session on advanced therapies, and I'm just going to hand straight over to Romina Aron-Badin, who's Associate Director of Molecular Imaging Research Centre in Paris. And Romina also, along with Professor Liam Gray, leads the Advanced Therapies Working Group.
Thank you, Anne, for the introduction, and thank you all for joining us in this fourth plenary session. So my first disclosure is that I am not in the clinical milieu. I'm a preclinical scientist, so many apologies if anything I'm saying here is not accurate enough for the clinic. But I was given the task of giving a brief introduction, an overview on the challenges in delivering therapeutics to the brain in times in your lives. And I thought in structuring my talk, trying to think of issues in a stepwise manner. So what we want to deliver is important, of course, where we want to put it in the brain, in which compartment, which cell population we're targeting, etc. Exhaustive enough. So in terms of the what, once again, the last few days have been enlightening in looking at the list that is always growing. But we started with drugs and some small molecules, large molecules, genes, cells, of course, antibodies, and any nucleic acids that can, or oligomers that can lower Huntington. And as I say, it's encouraging to see that the list is always growing. This might be delivered naked into the brain or might need vectors to get in. And we can use viruses, nanoparticles, liposomes, exosomes, vesicles of any sorts, scaffolds like hydrogels for delivering cells might be important, etc. And there again, we can see over the different days of the conference that the list is growing. So the combination of the two might have drawbacks in terms of target engagement and penetration. And we also have to consider factors inherent to the brain, like the permeability of the tissue that we're trying to get in, the diffusion coefficient, so biophysical properties of the substance, the therapeutic that we want to deliver that might work with us or against us, whether the therapeutic is metabolized in any way, whether it's recognized by the immune system, or whether there are clearance mechanisms that are activated by the therapeutic that we're delivering to the brain. So when we're thinking of where we start globally with the brain, and we cannot not talk about the blood-brain barrier, I don't think this audience needs me to explain what the blood-brain barrier is, just that it's so efficient in keeping pathogens and toxins out that it also makes our job really difficult in getting therapeutics. And the gymnastics of delivery get a little bit tortuous. So classically, we have used invasive approaches to get into the brain, which have may or may not have adverse events associated to it. We have to think, as I said before, of the immunogenicity of the therapeutics and the device and the approaches, but the first talk will deal with that in detail. The biodistribution of our therapeutic would also be affected by this. So do we want a local or a diffuse therapeutic? What is going to be more efficient? Can we engage with the target? Can we actually see that we have engaged to the target? Are there any off-target effects associated to it? And is the product modified in any way once it gets in? In terms of routes of administration, these will play a critical role to success. And classically, we've used intraparenchymal delivery, which is direct into the brain. It is invasive, as I've already said. It has risks associated to it, and it results in a local biodistribution most often. We can go into the CSF compartment via the ventricle, the cisterna magna, or endothelial. This remains invasive and has to face the high turnover, in particular if our therapeutic needs to circulate long enough to guarantee target engagement. It might be a problem and may result in a biodistribution gradient that renders our therapeutic less efficient. If we choose intravascular delivery, we're obviously less invasive, but we face the high turnover as well in the blood flow. Again, the BBB that I've mentioned before, and whatever is trapped or cleared in peripheral organs, which might make it inefficient, and there's little control of your biodistribution. And intranasal delivery is very easy access. The biodistribution will be dependent on the tracks where the therapeutics will migrate in the olfactory bulb via the trigeminal nerve, the CSF flow tracts, and there might be partial clearance in the mucosa as well. So these are just not negative points, but factors to consider when we're delivering, right? It's also worth mentioning that there are enhanced administration routes that are non-invasive, so MRI-guided focus ultrasound is a big delivery method that is coming into the clinic more and more. The idea, for those that are not familiar, is that we can deliver the therapeutic intravenously, coupled with microbubbles. These microbubbles will be sonicated with a high level of specificity. I don't know if you see my pointer there, oh, the flowers. So you can choose the region of the brain where you will localize this opening of the barrier by sonicating the microbubbles and rendering the BBB permeable to our therapeutics. In preclinical models, there has been a series of experiments that are proof of concept of different therapeutics that we can get into the brain. Gene therapy and cells are part of this, and of course, macromolecules and drugs. Today in the clinic, as far as I know, this has only been used for different reasons, like ablation for tremor, depression, or glioblastoma, but it's certainly going to be used for delivery of therapeutics and advanced therapeutics. In terms of devices, I'm not going to go into this a lot because obviously we have Liam Gray that will be talking about neurosurgical approaches, but just to know that even in the preclinical arena where we're facing delivering substances to the brain, we have a range of even catheter tips to choose from and that will determine how our product will be distributed, and it's very hard for us to predict, scale up, and imagine how this will work in the brain. And this will be determined by the design of our catheter, the type of injection rate that we choose, whether we're doing CAD or not. Is there going to be a single administration or a multiple administration that will be needed according to the therapeutics that we're delivering to the brain? What type of trajectory will be planned, etc. Just to give a peek onto things that are evolving fast in the field of devices, maybe. I just chose the soft implantable drug delivery device. It's not directly used for neurodegenerative diseases, but it just illustrates how the miniaturization of electrical circuitry can help in devising new deliveries and how the engineering is helping us in the preclinical and clinical arena. So this is a device that can be implanted in the brain to monitor electroencephalography, and this can trigger, if the signals are aberrant, the delivery of a drug subcutaneously. So one can think of how this can be generalized to other diseases. And in a similar vein, there's a couple of companies in the world, I am a member of the SAB of this one, so that's why I know about it, that are working on micro-robots. And this is, I would say, in the prodromal stages of development, if I may. It's very, very R&D for the time being, but the micro-robot exists, of course, and as you can see, as you can see down here, this is the micro-robot. It has the size of a grain of rice, and if you blow up, you can see that it has different units. These three are absolutely compulsory, and this is the one that you can change according to what you want to do in the brain. And the idea is to go from stereotactic frames to frameless surgery to surgeries that are less invasive, less time-consuming, and where you can maybe deliver substances to patients even in their room once the robots are inside. And the idea here on this structure is that you have the engine, the micro-engine and the propulsor for the robot to be able to navigate in the brain. Then there's all the electronics and the GPS in the third compartment to be able to guide and make the robot navigate externally with external sensors. And then the fourth compartment would allow you to deliver substances if there's a catheter attached to the reservoir. It could also do biofluid sampling locally in the brain somewhere, and it could also electrostimulate. So the idea is to have a device that is malleable and flexible to do different things in the brain. But again, this will have to stay tuned for a long time before it comes to the clinic, but it's just to show that things are evolving, and I think Liam next will show also what the prospects are in the clinic. So delivery per se is a challenge, but around delivery and what we deliver, there are other challenges that I will not in 15 minutes have the time to go into, like how you qualify the gene therapy product, how you acquire your preclinical safety and efficacy data, what do you consider to be efficacy, when do you consider that your product is ready to translate to the clinic, What are the pertinent readouts, follow-up, and endpoints that you can collect preclinically to help, once you get into the clinic, design intelligent clinical trial designs? And again, the first talk will show some of the clinical trial design evolution that we can expect. What are the outcome measures that we can use in the clinic, which will be tainted by what has been acquired in the preclinical arena as well, but not only? What are the regulatory aspects that we need to consider related to the therapeutic, but also to the device? And according to the agency, it might be both at the same time, as you know. And not least, what is the patient acceptability, the consent, the information that we give to them, the long-term follow-up, etc. All things that we have heard in the last few days were really, really important to the success of therapeutic translation. And if your favorite bottleneck is not listed here, again, I apologize for not being exhaustive, but just to say that these are some of the issues that we deal with in the Advanced Therapies Working Group, or we try to deal with and tackle, coupled with Stem Cells for HD, which is an international consortium. And if you would like to share your experience because you have ever overcome one of the bottlenecks and we don't know about it, or as I said, your favorite one is not there and you would like to share your experience with us, please, please, please don't hesitate to join. The doors are open, and we want to keep this dynamic together with Anne and with Liam in the surgical task force. And we organize task forces and working groups to try and issue papers and recommendations to help advance the translation of therapies to the clinic. So this brief overview was thought of trying to explain what the rationale was, together with Anne, to put up the program today. So having Dr. Liam Gray and Cheney from Cardiff University and the Cardiff Brain Center that will talk about what the innovation is in terms of neurosurgery and clinical trial design, respectively. And then there will be what is new in two different arenas of therapeutics, cell therapy and deep brain stimulation. We thought this could exemplify some of the bottlenecks that we have to face in the translation or in therapies that are already in the clinic. So as you know, and has been said before, Dr. Leslie Thompson could not join us today, but Anne has kindly accepted to give the talk for her. So she will be presenting the data. And because I'm sure that the speakers will keep up the time, we will have a lot of time for questions in the panel discussion. Save your questions for the panel discussion. We will just do all the presentations one after the other and have the questions at the end. So note them down and we will try to discuss later. So without further ado, my friend and colleague, Liam.